Dimorphic fungi are fascinating organisms with a unique ability to switch between two distinct forms, a mold phase and a yeast phase. This remarkable transformation is influenced by environmental factors, primarily temperature. In the environment, these fungi exist in their mold form, producing filamentous structures called hyphae. These hyphae can release infectious spores into the air, which can be inhaled by humans. Inside the human body, however, the warmer temperature triggers a transformation. The inhaled spores of dimorphic fungi convert into their yeast form, a single-celled budding structure that can multiply within tissues. This ability to switch between forms allows these fungi to survive in diverse environments and contributes to their ability to cause disease. Understanding the dimorphic nature of these fungi is crucial for proper diagnosis and treatment. Identifying both the mold and yeast phases, often through microscopic examination, is essential in clinical mycology. Coccidioides, a dimorphic fungus residing in soil, predominantly inhabits the desert southwest region of the United States. It presents a fascinating example of dimorphism, showcasing distinct characteristics in both its forms. In the environment, Coccidioides thrives in its mold form, developing hyphae that fragment into airborne arthroconidia. These barrel-shaped arthroconidia alternate with empty disjunctor cells on the microscope, creating a characteristic appearance. Arthroconidia production is an ingenious feature of certain organisms that avoids the need for fancy conidiophores and other structures. Instead, the conidia break down from the hyphae. These arthroconidia, highly resilient to environmental conditions, serve as the primary infectious particles, posing a risk of inhalation. Once inhaled into the warm, moist environment of the lungs, a dramatic transformation occurs. The arthroconidia of Coccidioides convert into spherules, the 37-degree form of this fungus. Because this phase of the organism does not grow in culture, it is not a true yeast. These spherules, significantly larger than the arthroconidia, continue to mature within the lungs. They undergo internal division, producing numerous endospores, further amplifying the fungal burden. In the mycology laboratory, these big bad dimorphs can create biohazards for lab scientists and students when not handled properly using a Class II biosafety cabinet. This biohazard is primarily a concern for lab scientists growing the mold form that produces the highly infectious arthroconidia. In this case, clinicians aren't in the same kind of danger because the spherule form is not easily transmitted. Way to go, mycologists! Taking one for the team. Coccidioides species, particularly Coccidioides immitis and Coccidioides posidacea, are the causative agents of coccidioidomycosis, commonly known as valley fever. This disease primarily affects the respiratory system, as the inhaled arthroconidia lodge in the lungs. While many individuals infected with coccidioides remain asymptomatic or experience mild flu-like symptoms, a significant proportion develop more severe manifestations. The clinical spectrum of coccidioidomycosis ranges from acute pneumonia, characterized by fever, cough, and chest pain, to chronic pulmonary disease with persistent symptoms and lung damage. In rare instances, coccidioides can disseminate from the lungs to other organs, leading to life-threatening complications. This dissemination often manifests as meningitis, bone and joint infections, and skin lesions. Section 4. Sporothrix, a worldwide dimorphic fungus. Sporothrix, another prominent dimorphic fungus, differs from coccidioides in its habitat and mode of transmission. Found worldwide, Sporothrix species is the last dimorphic fungus in this series that is found in the United States. It resides in soil, decaying vegetation, and even on plants like roses. Unlike coccidioides, which spreads through inhalation, Sporothrix infection, known as sporotrichosis, typically occurs through traumatic inoculation. The condition is also known as Rose Gardner's disease, and it is a condition true to its name. The fungus enters the body through breaks in the skin, often through pricks from thorns, splinters, or handling contaminated materials. Similar to coccidioides, Sporothrix exhibits distinct morphological forms depending on its environment. In its natural habitat, Sporothrix grows as a mold, producing delicate hyphae and conidia, the infectious particles. The conidiophores are very delicate, darken with age, and eventually form rosettes or flower-like conidiophores. The tiny conidia are attached to the conidiophore by invisible threads called denticles. These conidia, when introduced into the subcutaneous tissue through a wound, transform into the yeast phase. The yeast cells vary in shape from oval to round and also come in oblong forms known as cigar bodies. Sporothrix is the only organism among the American dimorphic fungi that easily converts into the yeast phase when grown on enriched media at 37 degrees. This feature can actually be used for conformation, a method known as yeast mold conversion. Other confirmatory methods such as DNA sequencing and MALDI are also available. Section 5. Conclusion. The importance of understanding dimorphic fungi. Understanding the dimorphic nature of fungi like Coccidioides and Sporothrix is paramount in medical mycology. Recognizing their distinct morphological forms, understanding their ecological niches, and appreciating their modes of transmission are crucial for accurate diagnosis, effective treatment, and implementing appropriate preventive measures. These fungi, while fascinating in their adaptability, pose significant health risks. As we delve deeper into the world of dimorphic fungi, we gain valuable insights into their pathogenesis, host-pathogen interactions, and the development of novel diagnostic and therapeutic strategies.